All right, so now we got to talk about primary, secondary, tertiary, and quaternary structure. So primary structure is the most basic. It's just the order or sequence of amino acids that you have. So order. So just the amino acids lined up together, and you can see it looks like a string like that. And there are, remember, peptide bonds. Don't forget that word, peptide bonds. Between each amino acid. That's what that red is. Peptide bond. Boom. Boom. You can see it's between this carbon and nitrogen. Okay. Remember, the amino group and the carboxyl group back from, I don't know, last year. Probably topic 2. Amino, I should know that off the top of my head. And carboxyl. Often we abbreviate it COOH, amino group NH3. And the R group, which is this right there, 20 different kinds of R groups. And that hydrogen is always there. And that control all subsequent levels of structure, subsequent all those secondary, tertiary, quaternary. And the chain of amino acids, that's called a polypeptide. There we go. Primary structure. Now on to secondary structure. All based on hydrogen bonds. Oops. And when we talk about secondary structure, we're talking about alpha helix, abbreviated that, beta pleated sheet. You might see a beta look like that. Yeah, here we have the alpha helix. Looks like a spiral. Spiral, almost like DNA. You're used to seeing DNA in that spiral staircase. And this is a beta pleated sheet. Kind of like you can make a, you make a fan out of paper, or some skirts are pleated. Pants used to be pleated. I used to have a nice collection of pleated pants, but haven't had that in a while. Look at pleated pants. They'll be back. Pleated pants. 10 years, I bet you pleated pants will be back in fashion. It all comes back. It all comes back. Oh, almost forgot. See these little, between the hydrogen and the H? Guess what? I knew you were thinking, Vandevort. Remember to label the hydrogen bonds, and I will do that. You won't have to email me or send me that Canvas. I like Canvas messages when you guys say hydrogen bonds. Not super strong, but the strongest intermolecular force. Remember, that is between molecules, not within. On to tertiary structure, that third level. That's when it folds up to make a more complex. We're getting more and more complex here. And remember the R groups, the 20 different R groups, the interactions between these R groups. And we'll see that hydrogen bonds, disulfide bridges, ionic bonds, hydrophobic interactions. And it is important for the function. Talk about structure leading to function. I transpose those two words. Maybe important. It is important. It is. Okay, I don't know why. And you'll see kind of like a spherical shape, kind of a glob. So that's why they're called globular proteins. Things like hemoglobin, things like enzymes, things like insulin, for example. So let's see, right here we have a hydrogen bond, right, between that hydrogen and that oxygen. And these are hydrophobic, hydrophilic interactions you see here. Hydrophobic, hydrophilic. Either way, that is having an effect on how huge effect on how proteins fold up. And for example, if you have a mutation and you have an area where you had a hydrophobic, like 2 hydrophobic amino acids, and then you switch, you mutate, and now it's hydrophilic, and instead of repelling, they then latch onto each other. That's going to cause probably a change in the DNA or a change in the protein that's going to have a serious effect. Then you'll see the S stands for sulfur, these disulfide bridges. Oh, for some reason I remember that disulfide bridge. For some reason. And then you have ionic. Oh, you chemistry. You guys that like chemistry, which I love chemistry too. You see the positive and negative. You'll know that's, ionic. Those are ionic bonds. Not terribly strong, depending, remember you chemistry people, depending on the difference in electronegativity. If you have a big difference, you are going to have a pretty strong bond right there. Okay, that's tertiary. Let's move right along to quaternary. You can see basically you're taking a bunch of different proteins and assembling them, sticking them together. So more than one polypeptide chain. Yeah, like it says. Assembled subunits. So those subunits are those different polypeptides. Remember, just starting out just the amino acids. Then you have the alpha helix or beta pleated sheet. Yeah. And then you have folding. And this is where most of the complexity arises here in this tertiary. I would say most important. This is where mostly you have that shape happen. And then, of course, it moves along, and those several polypeptide chains come together to make that completed protein. And, I don't remember. I gotta figure this out. I think from what I've seen, most proteins are, you have at least 2 subunits together. 2, 3, 4. Here you have 4. But I'm sure there's some finished proteins that are just one chain. But I'll look into that. I'll check that out. I love it. I always come up with questions I get to learn. All right, so here we have talking about specificity. Okay, so here we have a tRNA, and this is how we recharge, or how the tRNA is recharged. This is a tRNA-activating enzyme. So this right here, this would be enzyme. And it binds to ATP for energy and to a specific amino acid. Remember, these are, this is the way. Remember I said that tRNA, after it has its amino acid added to that chain, it pops off and then it gets recharged. So this is that recharging phase I told you about. So recharged transfer RNA basically is what I'm showing right here. So here is the enzyme. Oh, it got the energy. Oh, it has the amino acid. Now it's got to join with a specific transfer RNA that's specific to this amino acid. They must match. Must match up the amino acid and the tRNA. Must match. And then once that the energy, you have the phosphorylation, the dephosphorylation, loses that phosphorus, and the energy is used up, and you have that connected AMP then released, and then the charged tRNA is produced, recycled, all ready to go. And remember, this is going to go to the ribosome to make polypeptide. Woo! Jamming it right in there. Okay, so this is kind of cool. We have some images called polysomes. They look like beads if you look at through a electron micrograph. So here you have RNA and many different ribosomes attached to a single messenger RNA. So here we got ribosome there. So that's where we have ribosomes all attached, several. And this, you can see, this is the polypeptide being produced. And yeah, a little graphic right there showing. Okay, and this top one, this would be what it would look like for a prokaryote. This top one, and then this is eukaryote on the bottom. So this is eukaryotic, prokaryotic. So you see, it looks like it's a little denser. Yeah, this would be a eukaryote right here. Denser ribosomes, maybe occurring, maybe more efficient. Lots of proteins being produced at once. So they might ask you, probably not on a test, but they might ask you, it might come up, is this, again, like you see this is most likely a prokaryote, right? All right. Another close-up image. This is the direction of the translation. Remember, it's always 5' to 3'. Can't really tell from. That would be here. That would be there. 5 to 3. This is the ribosome. The little white, little teardrop thing. And that's the messenger RNA. Going right through all of these ribosomes. Oh, I made a mistake. Hold on. Remember, this is sorry about that. This is our DNA, and this is the messenger RNA is being produced when in autopilot mode. Sorry about that. This is actually RNA polymerase. Glad I caught that. Whew. So yeah, there we go. Whew. RNA polymerase producing that messenger RNA, then going into the ribosomes. And it doesn't really show it, but boom, this is where you're going to have the polypeptide, the amino acids, being produced. Woo! So here's we can use this molecular visualization software to look at these. These are the images that scientists have determined, figured out for these tRNAs, ribosomes, and the complex of both of them. Remember, there are 3 binding sites for tRNA, and I'll show you that in a second. 3. Really 2 exit sites. How that's really a binding site, because once that peptide bond is produced, it moves over and it kind of pops out. So this is what you'll see on your paper there. Let me go ahead and show you, and this is what you can go to this website and you can see the actual images, the actual structures. So here you can see the ribosome. This is that first one, I believe, just the ribosome. These are, you can see the structure of the proteins, the tertiary structure, and if it's a different color, it's a different protein. So it kind of shows you all the different proteins that are involved. Well, actually, that's not accurate. The different colors have to do with different chemical properties that scientists have discovered. But you can see, oh yeah, maybe zoom in a little bit. Oh yes, check it out. Oh yeah. And all the detail that scientists have come up for these. And I'm in the wrong view, but you usually can see also. How do I get that back? I reload it. Yeah, so you can see the sequence also. Sorry, there we go. I'm trying to scroll this. Come on, scroll. There we go. These are the actual corresponding letters of the gene that make up, of the messenger RNA. You see that, so U, A, C's, and G's, and I can actually click on these, and it'll take me right to where where that letter affects the protein structure. Pretty amazing. So scientists can use these tools to research, to figure out how this might interact. Any kind of protein with another protein, whether the drug, disorder, hopefully treat conditions. So this is a full ribosome. Let me now show you, this is a transfer RNA. You can see it has that kind of shape where it has a kind of column here and then kind of a side, like a little like a T, like with one side of the T chopped off. And you can see it's much smaller than the main ribosome. You kind of check it out, looking at that structure. And let's see. Now let's go to, oh yeah, this one I'll show you. So this is showing you, and maybe you can figure it out. What is this showing? Hopefully you can identify, see the kind of purple, the magenta, and this brown structure as tRNAs. So these are 3 transfer RNAs that are attached to a single ribosome. You can see the small subunit down here, large subunit up here. Now remember, this would only occur for just a split, tiny microsecond, nanosecond. Once you have that A site entering, that new amino acid, that new transfer RNA, you're going to pop out that old one that's already, given up its, polypeptide chain. But you can see, it's pretty cool. You can really see here that small subunit and the large. So when you open this up on your own computer, I really encourage you, to check these out and really spin it around. And there's a bunch of tools you can explore over here on the side.